Hi everyone, I am Dawn, a third-year graduate student researching electrochemistry in the Hub group. I respond to any pronoun but would like to be addressed directly by my name. I am from Guangdong province in China, a place very much like Florida in the US. Given up the warmth from the south, I went to the University of Minnesota for training in chemistry and sustaining the cold weather. Both experiences prepared myself well for what I am right now. So we have a concentration cell. Based on the color of the different components, we know that they are in different concentration. And this concentration cell is made out of the copper chloride salt with DI water added. And these two parts have similar concentration, while this one is way diluted than the other two. The first thing we are going to do is to turn on the multimeter to the corresponding reading. We want to read it in volts, and since the numbers will be small, we read it in millivolts. So right now you don't see a huge number, it's just fluctuating because we haven't connected the cell bridge to the cell, so the electrons essentially are not flowing and there's a break. Once we connect the break, it might also take a while for it to reach the desired number that it needs to be. You can see the number is increasing because it's trying to read what exactly the voltages are at this point. Once it reaches the equilibrium voltage, the number will actually start to slowly decrease as the concentration getting close within these two cells. Copper is it's as an electrode that is providing or getting the copper inside of the concentration cell. So if one side is doing the reduction, is going to receive the electrons and becomes like the ions in the cell is going to receive an electron, becomes copper metal, which will deposit on the side here. The other side is providing the electron, so the copper contact is acting as the copper source, and after the electron is gone, the copper on the electrode will be depleted into the solution, so that's why as the time goes by, the concentration of copper ion in the diluted cell will increase while the other one will decrease. So right now what I'm doing is to move one electrode or one end to the more similarly concentrated cell. The voltage that we read out of the cell is expected to decrease. The reading indicates whether you're connecting one side to the cathode or the anode. If you switch the sides, the sign will switch. So beforehand, we connect the black line to a less concentrated cell, so the voltage reads positive. And right now, we actually connect the black electrode to a more concentrated cell than this one. It's slightly more concentrated than what's connected to the red one, so the number is a negative number. But the voltage is not like, it tells you which side is anode and which side is cathode, but it doesn't affect the magnitude of the voltage that you get out. The cathode is the side that's doing the reduction. So if you understand it this way, reduction is taking electrons. So the ion has to become metal. Therefore, the more concentrated side is the cathode from the cell. Now that we've seen how the whole cell is working, we want to take a step back to see how each component of the whole cell was made. So this cell, as we can see, has 3 components that are pretty much identical. They are in different concentrations. To make each one of the components, the chemical that we are using is copper chloride. And to get a quantitative idea of what the cell is going on, you will need to measure the amount of copper chloride that's in the cell. But right now we are going to make a qualitative cell, so what we are going to do is to take a small amount of copper chloride, put it in the glassware, and then add DI water. And it is very important to use DI water or any water that is not full of different ions, because we know redox is the key in the cell. If we introduce other ions, the redox reaction will be different than what we expected. We need to put in the copper as our contact. You can use any copper, like copper plates, copper wire, or anything that's metallic. I'm using copper here because my metal ion in the cell is copper. So what I'm going to do is to just put it inside the glassware. So we pretty much finished one end of the battery. We usually call this one side or one half of the battery a half cell. So if we have 2 different cells and we combine together, it's a full cell. And remember the component that we mentioned before, a salt bridge? The basic idea is that you need to have a concentrated salt solution in between. And you can use a glassware or any types of tube-like structure. So here, I used a section of the tube that we found in our lab. And usually people will use a glassy, porous membrane that only allows ions to flow through. Here, to be more efficient, we use king wipe, where it's pretty common in the lab. You can find it in every lab. If you don't have king wipe and you want to make a salt bridge at home, use paper towel. And the solution that I was using inside this salt bridge particularly is saturated KCl. No particular reason that I'm using this one. It's because we use potassium chloride to store the commercially available reference cells. Therefore, we have a bottle available that's pretty handy. If you want to prepare your own salt bridge, you can use sodium chloride, sodium sulfate, or any type of salt that can flow